So when we look at growth, development, and genetics, so now here you'll see how, the uterus kind of gets ready to receive the fertilized ovum, and then how growth occurs, the various layers which form, and then, how you have a part which forms the fetal tissues, and then others which are shed off, like the placenta and amnion and so on, and then we'll do a little bit of genetics. So let's look at some terms that we use. Pregnancy is from fertilization to birth. That's obvious, everybody knows that. Conceptus is the developing offspring, so this should say offspring. The period of gestation is this development that occurs intrauterine. It differs for every species. In us, it's 9 months, and it can be calculated as 9 months. An easy way to calculate it is 9 months plus 7 days from something which is known as the last menstrual period.
Day 1 of the last menstrual?
Yes, it is this day 1 from the day 1 of the last menstrual period. So I know some animals have very short gestation periods, some animals have very, very long gestation periods, okay? So when we calculate it, we say it's called the expected date of delivery. The reason we say expected date of delivery is because of the baby is not born exactly on the day that you say it's going to be born. It could be born 10 days earlier, 10 days later. We always kind of give a range, and it's okay if it is born about a week to 10 days earlier or late. Beyond that or earlier than that, then you become a little bit worried. So it can be calculated as either if you know when fertilization occurred, which is very difficult to really know when fertilization occurred. So if you know exactly when fertilization occurred, you do 266 days after that. But otherwise, this 280 days after the last menstrual period, this is the one which I say is 9 months plus 7 days. And if you calculate, that comes to 280 days after the first day of the menstrual period. So if your first day is January 1st, then you will calculate 9 months plus add 7 days on January 1st, okay, with that. We have 2 types of development that occurs. All development does not occur only in utero, inside the uterus. So the part that occurs before birth inside the uterus is known as prenatal, but we have a lot of development that occurs after birth. For example, ossification occurs after birth. The alveoli of the lungs develop after birth. In fact, most of the development of the lungs occurs after birth. So a lot of postnatal development also occurs. What we are going to do here is just study a lot of the prenatal development. So early development is like this. So fertilization occurs. Then you can see at 1 week, the conceptus is this small. Then it becomes much larger. We begin to call it the embryo in the early periods till about the first few weeks we call it the embryo, and thereafter we start calling it the fetus. Okay. So let's say I give you a question like this. Eliza's last menstrual period was January 1st. When would you say her expected date of delivery is? Very good. October 8th. So I said January 1st was when she got her last menstrual period, and that was her first day. So you begin counting. So February, March, April, May, June, July, August, September, October. So October 1st she completes 9 months. Add 7 days to that. So that becomes October 8th. So that's how you will calculate. Okay? What happens at fertilization? There is a combination of the haploid gametes of the sperm and the ovum, so those chromosomes combine. Fertilization usually occurs in the ampulla, and when these 2 gametes fuse, we call it the zygote. The secondary oocyte lives only for 24 hours. and the sperm lives for about 24 to 48 hours. So the timing, if a couple wants to get pregnant, the timing of the intercourse becomes very, very important. Either a day before when you expect ovulation, or a day after when you expect ovulation. And therefore, if couples are finding it difficult to get pregnant, they always kind of wait and see when they expect their ovulation to occur using different methods. Remember, I told you that the sperms become fully motile only when they get into the female genital tract. So when the sperms are deposited in the female genital tract, a process called conditioning occurs. And here, the sperms they undergo what is known as capacitation. Their motility increases. They have kind of membranes covering them. Those membranes are removed so that they find it easier to swim. And that's why the female genital tract secretions are important to remove all of this. So when they do in vitro fertilization, then they actually have to add a particular type of fluid to allow all of this capacitation and conditioning to occur. So many million sperms, remember about 250 to 300 million sperms per ejaculate are deposited, but only about 50 or 100 actually make it to the uterine tube. The oocyte is actually surrounded by certain cells, and deep to those cells, so if you look at an oocyte, this is the secondary oocyte, it has a sort of ray of cells surrounding it which looks like a crown, so it's known as corona radiata. And then there is a membrane which surrounds the oocyte. That membrane is very important. That membrane is known as zona pellucida. This zona pellucida is kind of keeps this oocyte intact. It also prevents the oocyte from attaching itself to the fallopian tube. So when the sperm passes to fertilize, it will pass through this corona radiata, pass through the zona pellucida, and it reaches the secondary oocyte. Reaction, some enzymes are liberated. They cause digestion of the zona pellucida, and it allows the passage of the sperm. The moment one sperm passes in, this oocyte becomes impenetrable to other sperms, which is why at fertilization only one sperm is allowed to penetrate. If two sperms somehow manage to penetrate, then you'll have, 3 times the chromosome number will be triploid, right? Then that zygote will not be viable. It'll just die a natural death, which is good for us. So nature makes sure that you only have one sperm fertilizing an oocyte, okay? So as I said, the oocyte becomes impenetrable to other sperms. Also, secondary oocyte completes its second meiotic division only if it is fertilized. Here's an important thing. Within 24 to 48 hours after fertilization, a protein is produced, and this is present in the blood, and this is known as early pregnancy factor. So this is the way you can, if a woman thinks she's pregnant, so she tests her blood within 48 hours after the intercourse. When she thinks she's pregnant, she can check to see whether she's pregnant or not. Sex determination is done by the sperm, because the mother, in a female, the chromosomes are X44XX. So the oocyte will always have 22X when it divides. 22X, so this becomes 23 chromosomes. So there'll always be an X chromosome, whereas a male is 44XY. So when the sperms are formed, they can be either 22X or 22Y. So if the X meets the X, you get a girl. If the Y sperm meets an X, you get a boy. So it's the father who determines what the sex of the baby is going to be. Yeah.
What are the chromosomes if they have both? Like hermaphrodite?
Well, hermaphrodite is a bit different. In hermaphrodite, a true hermaphrodite is one in which you have both genital or both sets of reproductive organs being formed. So that they may be XY or they may be XX, but something went wrong during the reproductive phase and both genital organs got formed. So that's a bit different. You do have situations where somebody is born with XO. There are different syndromes. They have only one X. They don't have a second pair, Y or X, and that's called Turner syndrome. Or you might have a thing when they have XXY, so there are more chromosomes than normal. So those are different genetic problems that occur, okay? Now, twinning there occurs. There are two types of twinning. One is called fraternal twins. I told you all that ovulation occurs. You remember, opposite ovaries ovulate alternately at every month, and only one oocyte, secondary oocyte, is liberated at ovulation. Sometimes you may have more than one. Occasionally, you may have more than one. So suppose 2 oocytes were liberated, or maybe 3 oocytes were liberated. Remember, 50 to 100 sperms swim. So each one of those oocytes could be fertilized. So when they are fertilized, then you get what are known as fraternal twins, because the genetic material is not exactly the same. They could be different because different sperms have fertilized them. So they need not be of the same sex. So you could have a boy and a girl, you could have 2 boys or 2 girls, but they will not look identical. An identical twin is formed if after fertilization has occurred, the zygote will begin to divide. So first it divides into 2 cells and then 4 and so on. If each of these 2 cells after fertilization forms a complete human being, they will have identical genetic material. So identical twins will always be of the same sex and their genetic material will be identical. Okay? And that occurs after fertilization. Whereas fraternal occurs before. So many ova have 2 or more oocytes have been liberated at ovulation and they've got fertilized by separate sperms. So here this is the process showing you fertilization. So here you can see this is the zona pellucida, this is the corona radiata. These are sperms which are kind of trying to get in. The moment this is one sperm which has won the race, so those acrosome is on the top of the nucleus up here. It liberates enzymes which can dissolve the zona pellucida, pass through. The moment this passes through, all of these cannot enter. The zona pellucida becomes impenetrable. This has reached so far, but it will not be allowed to go inside and fuse with the female nucleus. Okay. So here this is showing you the same thing. So you can see here is the sperm nucleus. Here is the secondary oocyte which will complete its meiotic division. The sperm and the ovum, the nuclei swell. They come close to each other. When they swell, they are known as pronuclei. They approach each other and then they fuse. So now your chromosome numbers is chromosome and the amount of DNA is restored. So you become diploid again. And then this will begin to divide. So we can see here is a fertilized zygote. And this will begin to divide. So let's take a look at some questions up here. Why does only one sperm fertilize a secondary oocyte? The oocyte is permeable, as I said, to only one sperm. The moment one sperm enters, it becomes impermeable. It's not that the moment fertilization occurs, zygote begins division and others can't penetrate. It waits for a while for division to occur. So it's only because the oocyte is permeable to one sperm. So here, once that zygote is formed, it'll rest for a little while and then begin to divide. So even during division, the development starts. So we have 2 periods. The first is called the embryonic period, which is up to 8 weeks of development. This is a very, very important period because even though the embryo is very, very small, all your organ systems are beginning to form at this time. And this is the time when drugs and other substances which can cause changes and can interfere with development act. So drugs which cause harm to the embryo and they cause development to occur improperly are known as teratogen. And the way this was discovered actually is the teratogens was that in Germany they had a drug which was called thalidomide. And women were given thalidomide because during pregnancy they had morning sickness. And morning sickness usually occurs in most women during the early time, the early first few months of pregnancy. And it can be quite terrible. So they were given this thalidomide drug to, help them get over this morning sickness. they found that when the children were born, they were born with a lot of defects, congenital defects. One defect they were born with was known as phocomelia. You don't need to write this is just for interest's sake. So the children were born with just flippers for limbs, like this. So they didn't have, their limbs were not developed properly, and they were just little flippers. And they had other congenital defects, but this was kind of the most obvious that was seen. So they started studying and did a study, and they wondered, why was it that only these women had that, and others who had not taken the drug didn't have that? And that's how they started learning that development occurs during the first 8 weeks of, a lot of organ system formation occurs during the first 8 weeks. So if you start giving drugs during that time, that can affect development, which is why you will see that on many women, first of all, pregnant women are advised not to take any drugs during the first trimester, or at least the first 2 months, definitely not, because of this organ system development. The only thing that they are advised to take is folic acid, because folic acid prevents neural tube defects. But otherwise, they are told avoid as many drugs as possible. I mean, once in a while, if you take one day, you take Tylenol, nothing's going to happen. But over an extended period, they want you to avoid all drugs, because this is a very, very delicate period where you don't want to affect development, because anything can go wrong during this period. The next period is the fetal period, which is from 9 weeks to birth. Here, the organ systems actually begin to mature. So, you know, they grow, they get larger, they begin to secrete, urine begins to get formed, swallowing reflexes occur, all of these things happen. So that's the fetal period which occurs from 9 weeks to birth. So let's look at the early sequence of events. So when the zygote is formed, after the pronuclei have fused, it rests for a while, and then after about 36 hours of fertilization, it begins to divide. That division is known as cleavage. It's called cleavage and not just division, because the cells just get smaller and smaller. So if you have this big zygote, it will divide into 2, 2 will become 4, 4 will become 8, like this. So the cells start getting smaller and smaller. They don't kind of increase in size. So you just get a ball of cells by about day 3, maybe 16 to a few more cells. That ball of cells looks like a mulberry, so it's known as morula. At this time, it is traveling from the fallopian tube towards the uterus. So the zona pellucida kind of starts breaking down at this time. Some fluid from the fallopian tube enters into the morula, and so it pushes cells to one corner and forms a little sort of cavity there, and so that entire structure is known as the blastocyst. We look at the two parts of the blastocyst. You know, as I said, the cells are pushed to a corner. That area where they are formed, those cells are called the inner cell mass. They give rise to embryonic tissue. And then the outer layer is known as the trophoblast, and that gives rise to everything that is shed from the body. So the placenta and the fetal membranes. This blastocyst continues to travel through the fallopian tube, and it implants in the uterus by day 6 after fertilization. This is the reason why that zona pellucida is very, very important, because it takes about 6 days to travel through after fertilization. Fertilization occurs in the ampulla. It kind of rolls along due to the beating of the cilia of the fallopian tube, which set up currents, and they kind of push this embryo or the zygote through. So it takes about 6 days to actually reach the uterus. You don't want the zona pellucida to break down really early, because this trophoblast which will be formed is highly erosive. So it will can gnaw at the fallopian tube and attach there. So that's why it kind of breaks really, really slowly, so that by day 6, this blastocyst has reached the mouth of the uterus, and it goes into the uterus and it implants over there. Okay? In people who have some sort of problem with the fallopian tube, like a slight blockage or anything, you can understand this is when ectopic pregnancies can occur, because the blastocyst is kind of interrupted along its passageway. So at day 6, maybe it's still in the fallopian tube. It hasn't reached the uterus, so it will then begin to embed over there. So here you can see, so this is the oocyte which is liberated at the ampulla. Fertilization has occurred. It's passing through. So after about resting a little bit, the zygote divides into 2 cells, then the 2 become 4, 4 become 16 or more, you can see the cell size just gets smaller and smaller. So this is what is called a morula. As it is passing through the fallopian tube, a little bit of fluid gets in, and so it forms a little sort of cyst here. This entire structure is called the blastocyst. So it pushes cells to a corner up there. These cells up here are known as the inner cell mass, as you can see here. This is called the inner cell mass, which gives rise to the entire fetus, all the organs of the fetus. And this outer layer which is present here, this outer layer is known as the trophoblast, and that's what will give rise to the placenta and fetal membranes and help the fetus and embryo attach to the uterus. So you can see by the time it's kind of becoming the blastocyst, it's reached the uterus up here, so it makes it easy for it to embed. As I said, at this stage, from this it actually becomes 2 cells, 2 become 4. It's at this stage where if each of those individual cells formed an entire human being, that's when you get identical twins. So if these cells, each one decides to form individual human beings, you would get identical quadruplets. Okay. Okay, let's go back and let's look at implantation.
At the start of the menstrual cycle, a cohort of follicles starts to grow, but generally only one continues to develop. This follicle is referred to as the dominant follicle. In its interior, the follicle contains an immature oocyte. This follicle increases in size by 1 to 2 millimeters per day. The follicle produces estrogen and causes the lining of the uterus, the endometrium, to thicken. Around day 12 to 14 of the cycle, the follicle has grown to the size of about 21 millimeters. The rupture of the follicle ensues, which is called ovulation, and the mature ovule is released. The fallopian tube spin bag move towards the ovary and catch the oocyte that stays near the ampulary zone of the tube, waiting to be fertilized. If sexual intercourse occurs, the semen deposited in the vagina starts its ascension into the tube, and the sperm eventually arrive at the oocyte. One of these will succeed in penetrating into the interior of the oocyte. One day following fertilization, a cell with 2 nuclei is observed, at which point in time it is called an embryo, and it begins to divide itself. 2 days after fertilization, the embryo is made up of 2 to 4 cells and moves along the tube in the direction of the uterus. At the end of 4 days, the embryo continues to divide itself, at which point it is called the morula. And the interior of the embryo has an area filled with liquid, and this is called the blastocyst. The blastocyst migrates to the uterus and shapes the surrounding membrane, process of hatching, and the embryo begins to implant in the endometrium of the uterus.
So then that picture you saw towards the end, it showed how the trophoblast kind of sent out little projections, and that's how the beginning of the placenta actually starts. So let's see what, yes. Now, following implantation, let's look at how the placenta is formed. Remember, the outer layer of cells was called trophoblast, and that begins to form the primitive placenta. I told you that the trophoblast was highly erosive. So what it does is it kind of sends out little feelers and erodes the endometrium. And these little projections are known as villi, and they're known as chorionic villi, and primitive blood vessels begin to form in it, and these are fetal blood vessels. So when it begins to erode, it erodes the maternal sinuses of the endometrium, of the uterus. So these fetal blood vessels are bathed by maternal blood. At no point is there direct contact between fetal and maternal blood. There is separation by what is known as the placental barrier. Remember, it's called the placental membrane. Just like you had the filtration membrane when we did the urinary system, and you had the respiratory membrane when we did the respiratory system. So you have this kind of a barrier which kind of separates the blood inside the fetal blood vessels from the blood of the mother. Very few things can cross through that. Some of the things which can cross are drugs, which is why we don't want pregnant women to take medicines. The other things which can cross are those Rh antibodies. Remember, we said they were very small when we did blood. Those are the ones which can cross, but other very large molecules are not able to cross this placental barrier. The developing embryo, because that's why the secretory phase of the uterus was there, so that glycogen would be available, nutrition would be available, so that is done by the uterus. This trophoblast also secretes an enzyme called, a hormone, not enzyme, a hormone called human chorionic gonadotrophin, which goes into the blood, passed out in the urine, and this is the basis of pregnancy tests that you do using, what you buy from the store, and you test on the urine. You pee on the stick and see if you're pregnant or not. Enough is produced by the first to second week after fertilization. So that's why when you miss your period, you just miss your first period, you can test to see whether you're pregnant, because enough is produced by that time. The importance of this HCG is also that HCG stimulates the corpus luteum to continue to secrete progesterone, because progesterone is very, very important to maintain pregnancy. If, hypothetically, you removed the corpus luteum from the ovary after a woman got pregnant, the pregnancy would fail. I mean, she would abort immediately. After about 3 months, the placenta produces enough progesterone on its own, and that can maintain pregnancy. So after 3 months, you can remove the corpus luteum from the ovary and nothing would happen. But during the initial first 2-3 months, it's very important for that corpus luteum to continue to secrete progesterone to actually maintain pregnancy.
What happens to the corpus luteum? Does it always stay?
After 3 months, the corpus luteum begins to die off, because enough progesterone is being produced by the placenta. So the need for it to remain there is not there. Then, yeah, after pregnancy is over, and when she goes through a next cycle, then again the corpus luteum will form again. The cells, the trophoblast, apart from forming the placenta, it forms these what are known as fetal membranes called chorion and amnion. The amnion is a thin membrane which lines a cavity, which is actually what you probably know as the bag of waters. This is what is in everyday language called the bag of waters, which ruptures just before a baby is born. So it secretes this fluid called amniotic fluid, and some of the important functions of amniotic fluid and that amniotic sac, which kind of surrounds the baby. It's very much like, how you have the pericardial cavity surrounding the lung, or the peritoneal cavity, or pericardial cavity surrounding the heart, or pleural cavity surrounding the lung, or peritoneal cavity surrounding the various organs. Remember, they were protective. So this kind of forms like a water bath and encloses the fetus, provides protection. The fetus actually even swallows the liquid, and when the fetus passes urine, the wastes are removed and goes into the amniotic fluid. The external surface of the fetus, the skin, sheds into this amniotic fluid, and that's how when you do amniocentesis, you remove amniotic fluid, which has fetal cells in it, and you can see if there are chromosomal abnormalities. So that's what you do at that time. So that's what the trophoblast does. That inner cell mass, remember those cells which are kind of pushed to one corner, they are the ones which form the fetus. In the beginning, they're just like a clump of cells. Later, they begin to arrange themselves. So they arrange themselves in 2 layers, one on top and one below. So it forms a disc which has 2 layers. So this is known as bilaminar disc. After this, the bilaminar disc will turn into a trilaminar disc, forming 3 layers. And those 3 layers are known as, and you may have heard of it, we've talked of it often, those 3 layers which are finally formed are known as ectoderm, mesoderm, and endoderm. And these 3 layers are what give rise to everything in the fetus, all the structures in the fetus, going from skin right up to the lining of your inside cavities. So here let's look at how the placenta is formed. So here is the blastocyst which has embedded. So this is the trophoblast, and here is the inner cell mass. Notice how the inner cell mass has arranged itself in 2 layers. And then the trophoblast is kind of now sending out these little feelers which will go and rupture the blood vessels. So notice how it kind of begins to go into the maternal blood vessels and ruptures them. This is what forms those, what is known as the chorionic villi, which are part of the placenta. Inside this, blood vessels will grow, but they are separated by this placental membrane, so from fetal blood. So there is no kind of direct interaction between fetal and maternal blood. Meanwhile, this inner cell mass from 2 layers becomes 3 layers, and that's what forms the 3 germ. These are known as 3 germ layers because they give rise to all the fetal tissues, everything that's in your body, right from skin, as I said, to the inner lining of all of your organs. Here, as this grows bigger and bigger, so here you can see the placenta. So here are the maternal blood vessels. Notice how they pass into these sinuses. Here is the fetal side. So this is the fetal blood vessels, the umbilical arteries and veins, and they send branches inside each of those little villi. So notice this area up here where it is separated. This area up here, notice this area where this is what is formed, with what forms the placental membrane. So blood over here never comes in contact with blood over here. All it can do is substances can pass through this way, and only some substances can pass. But drugs are one thing which can pass, so we always tell a pregnant woman to avoid that. So this was at the placental level. Up here you can see that this is how a placenta has grown. So it's attached here to the uterus, connected to the growing embryo and fetus by this little cord, which was initially called the connecting stalk, and later it becomes called the umbilical cord. So here is the growing fetus which is formed by the inner cell mass. And here is where it is bathed by that amniotic cavity. In the beginning it's very small, but later it gets really large, as you can see here. So the fetus is actually swimming in this. You need a certain amount of amniotic fluid in order for the fetus to actually move about. If the fetus is not allowed to move, then the limbs can get stuck in a certain position, and then the fetus will be born like that. So you need this amniotic fluid not only to be protective, act like a shock absorber, remove waste, but also to allow for movements of the fetus. Here you can see this is the skin of the fetus. Notice how if the skin cells would be shed, they would go into the amniotic fluid. Therefore, if you pushed a needle through the uterus, through the amniotic sac into this fluid and withdrew that, you can actually test the fetal cells. So let's look at how amniocentesis is done.
Born a cancer specialist with a mission to provide personalized and comprehensive oncology.
Amniocentesis is a procedure that lets doctors detect or rule out problems with your baby's health long before birth. In the uterus, your baby is surrounded by amniotic fluid that provides protection and nourishment. Floating in the fluid are cells from your baby, providing doctors with genetic material for testing. In amniocentesis, your doctor will take a sample of this fluid to test for specific genetic disorders and birth defects. Amniocentesis is usually done in the second trimester of pregnancy at 15 to 18 weeks. It is generally offered to women with certain risk factors, age 35 or older, which increases the risk of chromosomal disorders such as Down syndrome, a previous child or pregnancy with a birth defect, blood test or ultrasound suggesting a birth defect such as Down syndrome or neural tube defects, or a family history of genetic disorders such as cystic fibrosis. Amniocentesis may also be done in the third trimester for these reasons: to determine if the baby's lungs are mature enough should early delivery become necessary, to diagnose a uterine infection, or to check for anemia in a baby with Rh incompatibility. To begin your procedure, your doctor will use ultrasound to create an image of the fetus and nearby structures on a computer screen. Looking at this image, the doctor can choose a safe place to insert the needle. Your doctor may give you an injection of local anesthesia to numb the surface of your abdomen where the amniocentesis needle is to be inserted, carefully watching the ultrasound monitor to avoid contact with the placenta or the baby. Your doctor will introduce the thin needle through your abdominal wall into your uterus and into the amniotic sac. He or she will then withdraw a small sample of amniotic fluid, remove the needle, and cover the site with a bandage. Your body will make additional fluid to replace what was removed. Your doctor will continue to monitor the ultrasound to check that the fetal heartbeat is normal and the baby tolerated the procedure well. You may feel mild cramping or pressure in your lower abdomen. For about 24 hours after your procedure, you will be advised to avoid physical stress, such as vigorous exercise, lifting, prolonged standing, and sexual activity.
So here, one of the things which I maybe mentioned earlier was, remember I said that the mother's age becomes important, and the reason was that females do not have any stem cells after birth. So if a woman has a baby when she's 24 years old, her oocyte is 24 years old, and it has been exposed to that much radiation and pollution and all of that. So the possibility of mutations would be much less. But when a woman is 35 years old or even 45 years old, then imagine that same oocyte is also 45 years old. So a lot of things could have happened to the oocyte. So the older the woman is, the more likely that there could be chromosomal abnormalities. Whereas a male, like I said, constantly producing stem cells, new stem cells with every batch, so their age is not as significant. Okay, so let's take a look at some questions. What is the structure that is responsible for maintaining the corpus luteum? What is that formed by? First think what the structure is, and then what is it formed by? The structure is the placenta, right? Remember, the placenta was formed by the trophoblast. The inner cell mass gives rise to the embryonic tissues, the fetal tissues. The trophoblast is the one which forms the placenta. So it is the placenta which maintains the corpus luteum, and that is formed by the trophoblast, okay? Which hormone does the trophoblast produce? That is a basis of pregnancy tests, urine pregnancy tests. HCG, yes, human chorionic gonadotropin. Early pregnancy factor is in blood, and that was formed within 24 to 48 hours after fertilization occurred. Estrogen and progesterone is by the ovary, not by the placenta, which the trophoblast forms. Okay, here. Very good. EPF in the blood, not in the urine, and HCG in the urine it'll be in the blood, but it'll be passed out in the urine. This is formed much later after the trophoblast has formed and the placenta has formed. So this is formed much later. Another question. No, there isn't. I just told you all. So I should have got 100 on this one. Somebody's not listening. Okay, so let's just look at this. So as I said, 3 germ layers are formed, and a flat, slipper-shaped disc is formed. When you are actually inside, when you're first developing, we are not like the way we are right now. So we don't have a cavity. So, we are not like this. No cavity inside us. What we are is actually a flat, slipper-shaped disc like this. So if you look at us from the top, we're absolutely flat, look like a slipper, no cavity at all. And this flat disc will have 3 layers. So if you kind of look at it from the side, it's like this: top layer, middle layer, and lower layer. And these are what are called germ layers. Here are some structures which you can see which develop from germ layers. And if you look at it, it makes sense. On the outside, what is present? Your epidermis and accessory organs of skin. The nervous tissue also actually develops from ectoderm. It's called neuroectoderm. Then just under your skin would be bone, cartilage, connective tissue, muscles. So all of these are formed by mesoderm. And then inside the cavities, the lining of all of your tracts, like your respiratory, GI tract, urinary bladder, and some other things like tonsils, parathyroid, and thyroid gland. These are some things which are derived from endoderm. So after these 3 germ layers are formed, the next stage, this stage was called gastrulation because 3 layers are formed. The next stage is when the nervous system starts developing. Nervous system starts developing very, very early, which is again one of the reasons why neural tube defects can be detected quite early, and which is why folic acid which is one of the things which prevents neural tube defects, which pregnant women are told to have. So that occurs first. And then the cardiovascular system starts developing by the 3rd week, and the heart begins to beat around 21 to 22 days. Now, I don't expect you to remember every kind of phase which occurs, so you must know what is given on this slide. In other words, that the heart begins to develop around by the 3rd week, and it begins to beat by 21 to 22 days, which is why when they do an ultrasound, they can see this primitive heart beating. What the 3 germ layers are, what are some of the derivatives of the 3 germ layers. Okay, so this is what I want. So let's look at the embryonic period. As I said earlier, very, very important because all the organ systems are forming up here, but they don't begin to really function too much at this time. The embryo folds. Remember I told you that initially the embryo was flat. It was like a flat slipper. So if you looked at it, it was just like a flat slipper like this, okay? Then it begins to fold. So it folds on the top and the bottom. So these are known as the head and tail folds. It begins to fold on the sides as well. So now if you took a section through the embryo, say from the middle, you will see the fold forming like this, okay? And that's how a little tube will be formed in the center, which becomes the gut tube, okay, or which will form the gastrointestinal tract. This is also the time when the limb buds begin to appear. So in the beginning, they appear as tiny little buds, which is how, they found in that thalidomide incident that the buds just remained as buds and they didn't grow further. The upper limb buds come up faster than the lower limb buds. The brain and the face develop much faster. If you remember when we did the cardiovascular system, I told you how the pulmonary artery, the trunk, was connected to the arch of the aorta by means of the ductus arteriosus, if you remember. So here is the arch of the aorta, and here are the 3 major branches which go to the head, neck, and upper limb. And here is the pulmonary trunk, and there's this ductus arteriosus which connects them. And this is after the subclavian artery. So this pulmonary artery is bringing deoxygenated blood, and through this it goes into the arch of the aorta. So the upper part, all these get more oxygenated blood, but the lower part of the body gets less oxygenated blood. With the result, the brain and head develop much faster. That's why the child's head looks really big. The fetal head looks really big, and by the time the child is born. Also, limb movements, the limbs, because of the presence of the amniotic fluid, the limbs begin to move a little bit. Around the 8th week, the mother can't feel it at this time, but little bit they begin to move, and that's why you need adequate amniotic fluid to allow for these limb movements to start beginning. Urine production, the kidneys start functioning by about the 8th week, so actually the urine is produced, goes into the amniotic fluid, and the fetus actually swallows it. So, kind of to try to keep it constant. If, for example, the nervous system is not developed properly, then the fetus can't swallow because the nervous system controls swallowing, the swallowing reflex. So then what will happen is that you will keep producing amniotic fluid, plus the fetus will be urinating, which will pass into the amniotic cavity, and the woman will have more than normal amniotic fluid, which is known as hydroamnios, because now the baby's not swallowing that. So that's one way of actually keeping it constant. Or if the kidneys are not functioning, let's say there's a maldevelopment of the kidneys, they don't function, what will happen? Amniotic fluid will be a little less than normal, okay? So that could lead to other problems as well. So you can see that everything has to be functioning properly. I also mentioned that teratogens were the ones that acted during this time, mainly at this time, and I gave you the example of thalidomide. Let's take a quick look at some questions. So what is one way to keep amniotic fluid constant? Yes, very good. Fetus swallows it. And I showed you the video on amniocentesis. So what are you looking for when you do an amniocentesis? Fetal cells, yes, remember the skin of the fetus is bathed by the amniotic fluid, and just like us, the fetus also sheds the skin. The cells are there in the fluid, and you pull those cells out with the fluid, and then you test for chromosomal abnormalities or other things in that, okay? Then after the embryonic period, we come to the fetal period, which is from the ninth week onwards. This is where the organ systems are beginning to grow. They grow in size. Also, fetal movements now occur quite vigorously, and these are felt by the mother. This is known as quickening. It occurs anywhere between 17 to 20 weeks of pregnancy. During the first time a woman is pregnant, she will not feel the fetal movements until around 20 weeks. But second time onwards, she knows what to expect, so she feels it a bit earlier. So that's why we have this range of 17 to 20 weeks. Surfactant production begins around 24 weeks, just keeps increasing after that. So, you know, once surfactant production begins, the fetus really becomes viable. Though after the if the fetus is born too prematurely, you have to put the fetus in an incubator and maybe even give extraneous surfactant to allow the fetus to breathe. Growth is, of course, rapid during this time. So here is a quick view of how you change from a flat slipper-shaped disc to somebody with a cavity. So here is showing you the two germ layers, and that's the amniotic sac, and this is called the yolk sac. And then here, a third germ layer comes the mesoderm, which is in between ectoderm and endoderm. So you're really flat at this point up here. So this is if you were looking at it from the top, okay? So it's showing you the way the mesoderm kind of intervenes and comes in between the ectoderm and the endoderm below. So it comes in between. And this is how folding occurs. So folding occurs on the top, at the bottom, and on the side. So this is just showing you the lateral folding. Here you can see the tail fold and the head fold. So you can see a whole kind of cavity begins to form, and that will form what is the primitive gut tube. So let's take a quick look at this.
In this video then we'll look at the beginning of development, looking into the ovary and an unfertilized egg. It then gets fertilized by sperm, which travel down the fallopian tube. So here, millions of sperm coming along. Several of them will hit the egg and try to penetrate it, but one will win, as it were, go into the nucleus, And then there's a reprogramming process where the male and female nuclei have their genes set aside to be turned on and off for early development. Here you see early cleavage stages occurring, and this is one of the early growth phases. As the embryo moves down the fallopian tube, it's going to form an important stage called the blastocyst here in a few seconds. Of course, in real life that takes days, about 5 days. At this stage then, I'd like to draw your attention to the inside of the blastocyst where there are cells called the inner cell mass, which I'll be abbreviating as ICM. Those are the cells that make the entire animal. And the outer cells give rise to the placenta and other supporting tissues. At this stage, the embryo implants into the wall of the uterus. This is when a pregnancy is really initiated. And now we'll see those blue inner cell mass cells form a disc, and then as the cells continue to grow, they change their physical positions, their kind of geographical relationship to one another. And you'll see that represented here as this disc gets transformed into an embryo. Those lines represent sites where cells are migrating in and out. And here's an important stage when the 3 beginning layers of the embryo, the so-called germ layers, are formed, and I'll come back to that in a few minutes. As development proceeds, there's more growth and movement of cells. It'll begin to form a neural tube. Here it turns, and appendages start to bud out. You see the head forming and the eye, and then eventually we get a small embryo, and some months later, of course, this would be born as a young baby.
So that was really quick, but you saw how it, from a flat disc, how you kind of got a cavity inside. So this is showing you the same thing. So if you look at a 8-week embryo, perfectly formed, just very, very small, perfectly formed. And you can see a 12-week embryo is also pretty small, but you can see everything visible in that. So this is just kind of showing you the stages and approximate sizes relative to each other. So we talked of twins. So let's take a look at this question. What's the difference between fraternal and identical twins? False. Yes, it's identical who have to be of the same sex. Fraternal may or may not be of the same sex. Let's see some changes that occur during pregnancy. First is there is cessation of menstruation. So there is no menstruation occurring at this time because the corpus luteum survives. It's going to continue to produce for the first 3 months progesterone, so the endometrium continues to build and nourish the embryo. So whether pregnancy occurs in the fallopian tube, which is ectopic, or ovary, which again is ectopic, or anywhere else, which may be ectopic, or it occurs in the uterus, you'll always have cessation of menstruation. That period is known as no menstruation, so it's known as amenorrhea. Pregnancy is approximately about, we say 38 weeks to 40 weeks. It has 3 trimesters, okay? The first is the first 3 months, second is the 4th, 5th, and 6th, and the last trimester is the 7th to the 9th month. The uterus is one organ which physiologically is capable of increasing its number of muscle fibers, which is known as hyperplasia. So not only does the size of the uterus obviously increase, which means the individual muscle fibers increase in size, but the number of muscle fibers also increase. Normally, in no other part of the body, physiologically, do the number of muscle fibers increase. When you increase your muscle mass, only the size increases. But physiologically, the uterus, the number also increases, which is why after pregnancy, the uterus, when it what is called involutes, it tries to come back to its original size. It can never come back to its pre-gravid state because you see the number of muscle fibers has increased, so it will always be larger than before the person got pregnant. Oxytocin receptors also increase during pregnancy naturally so that they can react to the oxytocin which will be liberated during labor. Certain other things happen. Some women have deep pigmentation around the facial skin. That's called chloasma. That doesn't go away. Others are lucky, don't have as much. The glandular part of the mammary glands increases. The placenta produces this hormones called relaxin, which relaxes the ligaments, which is why there is a slight degree of movement which is present between the pubic symphysis and even at the sacroiliac joints. It relaxes them, gives women, pregnant women, the typical waddling gait. This also helps so that during labor, the pubic symphysis can separate a little bit and make the birth passage a bit longer, bigger. So that's important. Weight gain is seen. Most systems are affected by pregnancy, and the process of childbirth is called parturition. So let's see what happens during parturition. So we have 3 stages of labor or parturition. The stage 1 is from when the uterine contractions begin till when there is complete dilation of the cervix. Remember, the cervix was closed. It was a closed canal like this, right? And this os was there. In complete dilation, this is what it will look like. So this is entire, the cervix is dilated, usually about 10 centimeters, so that the fetal head can, and the rest of the fetus can pass through. This is the longest period. This is when which is most painful. Because of the contractions, the fetus within the amniotic sac is pushed down. So when the OBGYN feels pervaginally, they usually feel the bag of waters, and that's usually the leading part, and that ruptures and helps to push the baby down. The second stage of parturition is called when the fetus is completely expelled. So from the stage of complete cervical dilatation to exit of the fetus. And then the last stage is when the placenta is pushed out. So here if we look at it, this is in one. So notice how the cervix is closed up here. So now the cervix is beginning to dilate. So it will kind of really be pushed way beyond so that this whole thing opens up like you can see up here. So here is the bag of waters. Sometimes it doesn't rupture on its own, and then they just have to give it a gentle poke and rupture it so that all the water comes out, and then the uterus kind of contracts directly on the fetus and helps to kind of push it out. Here you can see stage 2, the fetus coming out, and finally the placenta comes out. The obstetrician has to make sure that every part of the placenta comes out. They always examine the placenta very, very carefully, because even if a little bit is left behind, it prevents, once the placenta comes out, the uterus begins to contract again so that it closes off the maternal sinuses. If even a little bit is left behind, any tissue left behind, it will prevent the uterus from closing, and the woman will continue to bleed postpartum, which is called postpartum hemorrhage. So they always check to see that the placenta is whole and no bit is kind of missing. Okay, we'll now move on to genetics. And we'll talk about heredity, which is how traits. Traits are nothing but characteristics, like having blonde hair, green eyes, being tall, short, flat feet, dimples, freckles. All these are traits. So those are traits, are some things you can put in, which you can use as an adjective. Okay. So heredity is just passing of traits, and how these traits are passed are by the presence of genes. So genes carry these unique traits, and they pass it on from the parent to the child. Genes are found on chromosomes. Humans have 46 chromosomes, or what we call 23 pairs. Of these 23 pairs, we say that 22 pairs are called autosomes. So 22 means we can say 44, okay? And the last pair, so one pair is the sex chromosome, a pair of sex chromosomes. So that's how you have 23 pairs. Now, if I unravel the 23 pairs and represented it as 46 chromosomes, here you'll have 44 autosomes and 2 sex chromosomes. If it's a woman, it will be XX. If it's a male, it will be XY, and those are the symbols for male and female. What genes do is they control the production of proteins, which is what determines what traits you will have. Okay, so that's a function of gene, and only one specific protein is controlled by each gene. There are some genes, not everything is on a dominant recessive basis, but for certain traits to be passed, there are some genes which are extremely dominant, others which are weak. And a good example would be height, for example. If someone is really, the tall gene is a dominant gene, the short gene is a recessive gene, okay? So dominant genes mask recessive genes. So whenever you have, like, because all these chromosomes are present in pairs, whenever you have one dominant gene and one recessive gene, the trait will still be shown because the dominant masks the recessive. So you don't necessarily have to have both dominant genes in order for the trait to be expressed. But in order for a recessive trait to be expressed, then you automatically have to have both of the recessive genes, because you cannot have anything masking it, okay? So that's an important thing. The other thing is, traditionally, whenever we talk of dominant genes, we always represent it in uppercase. And just to make it easier, we always write the dominant gene in front of the recessive gene. So, for example, if I want to say somebody is tall and I write it like this, they have only one dominant gene and one recessive gene, and let's say the gene for height was represented by T, we traditionally write it like this. You can write it this way, that's not wrong. It just makes it a bit difficult to understand at first place. We always put the dominant gene in front, so immediately you can see that this person will be tall. Okay, that's just a traditional way of writing things. Alleles are something which are the forms of the gene, the traits. So these two are what are called alleles. So this is the allele, tall allele, and this is the short allele, for example. So they are basically just the forms of the gene. So for height, there is a tall gene and a short gene, right? So this is the tall allele and this is the short allele. Usually, for us to inherit a trait, we always have to have two of the alleles. So we can have a big T and a small t. We can have two big T's, or we can have two little t's. That means we get both the recessive genes. Here we get both the dominant genes. Here we get one dominant and one recessive. In either case, can you see we are getting two alleles, okay? There are certain traits which do not go by just these two alleles. You have multiple alleles. Skin color is one of them, because skin color has many alleles, and depending on the amount of pigment that is present, that gives rise to your skin color. Often people tend to think if a very dark person marries an extremely fair person, usually the child is in between. They kind of, like an in-between color, and that's just because you mix the two and then you get an in-between. That's not how it works. It looks like that, but that's not how it works. It depends on how the melanin, how these alleles have kind of aligned with each other, which you can see that there may be parents who maybe one person may be very dark and another person may be very fair, and let's say if they have 3 children, you can see actually the 3 children maybe of their complexions may be a bit different, okay, and that's because of this multiple alleles. And we'll see blood groups also have different alleles. Now, when the alleles are same, we use the word homozygous. So here you can see the two are same, the alleles are the same, big T, so this is homozygous. These two are same, this is also homozygous. We would call this homozygous dominant, because both are dominant alleles. We would call this homozygous recessive, because both alleles are the recessive type, okay? When the two are different, we call it heterozygous. So this would be heterozygous. Now in this, we also have two terms which you have to be very conversant with. You must know what they mean. One is called the phenotype. That means the physical makeup. So when you look at a person and you can actually describe them, so this is what is tangible, you can actually describe them using an adjective. You can say this person is tall, this person is short, this person has freckles, something like that. Then you're describing what is called the phenotype, okay? So, for example, as I said, a dominant gene masks a recessive gene, and you need to have only one of the dominant allele in order for that trait to be expressed. This person will also be tall, right? And this person will also be tall. But by just looking at someone and we say that person is tall, we don't know whether their makeup is this or their makeup is this. We cannot tell, right? So when we just describe the person, what we are describing is their phenotype, so that physical makeup. The genotype is actually how you would express it genetically. So in order to do that, you really have to do the genetic study. So this would be a genotype, this would be a genotype, this would be a genotype as well. Got that? But you cannot see by looking at a person what their genotype is. So that's what the genotype means. So let's review. How many pairs of autosomes are there? Very good. Okay, we'll look at patterns of inheritance. So there is one which is the kind I just described, which is called complete inheritance. So no gray areas. You're either dominant or you're recessive. So the alleles are expressed as dominant and recessive. And I told you that recessive have to be homozygous. They cannot have anybody masking them. So therefore, they have to be homozygous in order to be expressed. Whereas the dominant can be homozygous or they can be heterozygous. Then we have something which is known as incomplete inheritance. In incomplete inheritance, there's kind of an intermediate form. Neither one is dominant. And so you kind of see an in-between thing up here. You see it in plants. So, for example, if you had a red flower and a white flower and you mix the two together, you get a pink flower. So that's incomplete. So you get something in between. We'll see the Palomino horses. There's a white and a brown horse, and when you mix them together, the part of the horse is brown, part of the horse is white. Like their tail is white and the body is brown, or the mane is brown. I forget which is which, but they are like that. But they are because of a brown and a white horse having been mixed together. Incomplete inheritance, many books put sickle cell anemia in this, others do it in codominance. I'm using kind of your book. It's also seen in sickle cell anemia where it's incomplete in the sense that a person will either have a milder form of the disease if only one of the genes is expressed, and that is called the trait, or they may have the full-blown form of the disease when they have both of those genes together. Okay, so we'll see that. We'll see how this happens. Then we have another kind of inheritance which is known as codominance, where let's say there are two genes and neither one of them wants to give way to the other. So both are expressed. Blood group is one such example. For example, the A antigen and the B antigen are both equally dominant, but the O is actually recessive. So if A and B are present together, you get AB blood group. If A is present with the O allele, you'll get A because O is the recessive one. If A is present with A, you obviously still get A. So we'll look at this too. Okay, so we'll talk about codominance. And then we'll talk about sex-linked inheritance. Most sex-linked diseases are carried on the X chromosome. The only known sex-linked trait which is carried on the Y chromosome is a hairy external ear, hairy pinna. Some men, the Y chromosome comes from the father. So some men inherit that where their ears are extremely hairy. So that's the only known one that travels with the Y, the known one that I know of. You all can research and see if there's any others. I haven't been able to find anything really much. We'll also see, and in sex-linked diseases, we also talk of a state which is known as the carrier state. And here's a thing you have to be very clear about. you see, when in other diseases, when you have in other all of these, or what are called autosomal traits, in all of these other diseases, when you carry a gene, you may manifest the disease, then you automatically say, yeah, you're carrying the gene. That's obvious. Unless you carry the gene, how can you manifest the state, right? But in sex-linked, we will see how a woman can carry the gene, and when she is known as a carrier, she's just carrying the gene, but she does not have the disease. So this carrier state is very, when we call it the carrier state, this is very typical to sex-linked diseases. And I'll show you how this happens, okay? So here is, you have these, so you can follow along with me. So let's look at how we determine sex, or whether it's going to be a child, or whether it's going to be a boy, or whether it's going to be a girl. So during oogenesis, the oogonium will divide and it will form, you know, 20, remember it will form ova, right? 4 cells which it will form. All of the 4 cells will be 22 plus X, right? Because a female has 44 plus X. If you divide 44XX, if you divide it right down the middle, you'll get 22 and X, right? So she'll always just have an X chromosome. So we will write, it will break up into X and X. That's all she can have, X and X. In the case of the guy, he is 44XY, right? So he can get 22 plus X, or he could have a 22 plus Y. So his chromosomes, the sperms could carry an X chromosome, or they could carry a Y chromosome. So therefore, in the male, we don't know whether it's an X, sperm carrying X or Y. So we'll write X or Y, depending on which one will meet the female. So here, what we do, this thing is called a Punnett square. So during fertilization, this can meet with this, and you can get XX. This can meet with this, you get XX. This X meets with this, you get XY. This could meet with this, you get XY. So out of 4 possibilities, you can see that there is 50% chance of getting a female, 50% chance of getting a male. Okay? These are the 4 possibilities that you have. So in any the difference is usually statistically when you see it's usually around equal. The timing matters a bit. If sometimes if the intercourse happens just before ovulation or just after ovulation, sometimes it favors one particular set of sperms. So, you know, other than that, but otherwise you can see theoretically the chances of getting a male or a female are equal. Now let's look at incomplete inheritance. So first I wanted to show you this so that when we do sex inheritance, we'll be able to work that out. So now when we do autosomal traits, we'll forget about the sex and we'll just break it up into what trait we are talking about. So let's say that if someone had to have dimples, so we are talking about the trait dimples, and I tell you that the gene is dominant and we represent it by the letter D, big D. So in order, if you have dimples, you have to have at least one of the big Ds. And if you don't have dimples, you'll have to have the little Ds. Then I give you this, that the parents are heterozygous. So if both parents are heterozygous, that means both parents have to have a big D and a little d, right? Got that? So now, when the gamete formation, this is how the mothers will break up. You'll break it down the middle. This is how the fathers will break up down the middle, okay? So, this and this meet, you will get this. 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 So, have you followed that? What happens? So, in this, out of 100%, this person will have dimples. This person will also have dimples, as will this person. This one will not, right? So what are the chances that a child will not have dimples? Very good. In a pregnancy, the chances that a child will not have dimples is 25%, because we take 4, so this is 25%. The chances that a child will have dimples is 75%, if both parents were like this. Okay? So this is complete inheritance. So that's how you will draw the Punnett square and you will work it out. So you might, in your test, you might get something like this, and you can work out what will happen. Let's look at incomplete dominance, where you kind of get an in-between thing. Okay, so I'm using the horse. We could use flowers. So here, this is a brown horse, and this is a white horse. So when they mate, we'll get everything which will be the same. 100%. Any pregnancy, you will always get a palomino horse which will have part brown or part white. Okay. Instead of this, if I used, let's say, a red flower, and we say red is R, white is W, and pink is RW. And this was white, so white can remain as such. So this is red, white can remain. So when you cross a red flower and a white flower, you will always get pink flowers. Do you see that? Okay, so this is incomplete. Neither one is expressed, you get something in between. Let's see sickle cell disease. The normal hemoglobin is represented as S. The sickle cell anemia, the gene for sickle cell anemia is represented as S dash. Okay? In order to have the full-blown disease, it should be like this. In order to have a milder form of the disease, which is called the trait, you have only one of the S's. Okay? And here I'm giving you this example that both parents have the trait, right? Can you see this? Both parents have the trait. So this is how it will break up. So if this meets with this meets with this and this meet, and this meet. So what would be the phenotype of this person? This one. Normal, right? This person would be normal. What about this one? Trait. Don't say carrier. Trait. Don't use the word carrier here. Trait. They have the trait. Okay? What about this one? This one is also trait. And this one? Full-blown disease. Okay. So we'll say 25% genotypically, 25% are normal, 50% have the trait, and 25% have the full-blown disease. Right? Okay? Let's look at codominance. So here, A and B are both equally dominant. So these are equally dominant. A and B, the genes carrying the A and B antigens are both equally dominant. There's no O antigen, but the gene for the O group is recessive. So therefore, since you have to have two alleles, so this is a bit of codominance and a bit of dominant recessive also. You can see it's a mixture. So since you have to have two alleles, if you are blood group A, you can be both A's, or you can be an A with a little O. Okay? And if you are blood group AB, obviously you'll be like this. If you are blood group O, because O is recessive, you have to have both O's together. Okay? Some books will, instead of doing it as an O, they'll do it as an I. Just not to confuse you, because of the codominance, I'm not putting the O as a little o, because recessive, remember, traditionally we write a little, I'm just writing it as a big O. So let's look at this situation where I give you one parent is heterozygous A and the other parent is homozygous B. So you should be able to recognize that. So if that parent is heterozygous A, how would you write that? AO, right? And if they're homozygous B, that means you've got to write BB this way. So when we break them up, this is how they will break up. So let us see what the possible blood types are. This and this will meet, you'll get AB. This and this meet is this. This and this meet, you get this. And this meet, you get this, right? So the possible blood types of the children would be either the child would be AB or the child is B, right? So we all know that the mother is a certainty. She's the one who produces the child, right? So the mother is a certainty. Father is where it gets a bit iffy if you're kind of wondering about paternity. This is a quick way to test paternity. You can prove that somebody is not the father. Because if, say, a couple, the mother is A and the father is O, right? And they get a child who's O group, right? Then you know that that this guy is not the father, right? Because, let's say the mother is A and the father is this B and the child is born as O. Then you know that this person is not the father, right? So that you can only tell if this person's not the father. You can't tell for certainty that this is the father, okay? Yes. Now let's take another situation if both parents are heterozygous. So this one is also heterozygous. So this here is BO. Yes, then they can. So this one becomes AB, this one becomes BO, this one becomes AO, and this becomes O. Then they can have. Right. Okay. So you have to work out the permutations and combinations based on what the genotype of the parents are. Yeah, not the phenotype. You have to know the genotype. Yeah. So here let's look at sex-linked inheritance. So for hemophilia, which is due to lack of a clotting factor, there are different types of hemophilia. So there's either clotting factor 8 or 9 is absent. So let's say the normal gene for hemoglobin is H, and the hemophilic gene, let's say we represent as little h. Okay, so not for hemoglobin. The normal gene for normal clotting is H and the hemophilic gene is little h. And then I tell you that the father is hemophilic and the mother is normal. Okay, hemophilia is carried on, the gene for hemophilia is carried on the X chromosome, not on the Y chromosome. The Y chromosome does not have any gene attached to it. So therefore, let's look at it. So when we did, remember when we did just sex determination, we had, if I draw a square quickly, so the mother's alleles will be X and X and the father will be X and Y. Okay? Now here, as I said, the gene for hemophilia or even the normal gene is only carried on the X chromosome. The Y chromosome doesn't have anything for hemophilia or normal attached to it. So I'm telling you that the mother is normal. So on both her X chromosomes, she's carrying this normal gene. Okay? The father, on the other hand, is hemophilic, which I tell you that on his X chromosome, he's carrying this gene. And on the Y, he has nothing, because the Y doesn't carry the allele for it. So let's see the possibilities. So this would be X and X, like this. This and this meet. Okay. Right? So there's 50% chance it's going to be a girl, 50% chance it's going to be a boy. If a boy is born, the boy will be normal. Can you see? Okay. If a girl is born, because remember, this is the normal gene, this masks this recessive gene, the girl is going to be normal, but she will be what we say a carrier. She will not suffer from the disease, but she will carry the gene. So she can actually pass it on to her children. So let's look at such a case. So instead of this, suppose I said the mother is a carrier. If I gave you this state where the mother is a carrier, okay? So then here it will be like this. The mother is a carrier and let's say the father was normal. So if I give you that the father is normal and the mother is a carrier. So this is how the mother will be. So here Okay? So here you can see normal girl, carrier girl, normal boy, hemophilic boy. See, he doesn't have another X chromosome to protect him. So when he gets the gene, he suffers from it. In her case, both of her X chromosomes have to have the hemophilic gene in order for her to suffer from hemophilia. Right? So she's a carrier. Notice she is the one who passed it on to the boy. The mother always passes it on to the son. In order for a woman to have the disease, then the father should be like this, and let's say her mother is also like, is a carrier. Her mother is a carrier, father suffers from the disease. So now you can see this is the possibility. So this one is a carrier. She suffers from the disease because she's got both of these little H's. This guy is normal. This guy suffers from the disease. So in order for a woman to carry it, a woman to suffer from the disease, she should get it from both parents. Okay? She should get the gene from both parents. Got that? So, why do you think only the mother passes on the X-linked diseases to the son? Yes, very good. The son only gets his X chromosome from the mother. The father has X and Y, and a boy is X and Y. The Y has to come from the father. The mother can only give X chromosomes. So the son gets his X chromosome only from the mother. Okay, that's the reason why X-linked diseases are passed on to the son from the mother. Yes, and this is what we did. She has to get it from both parents. This is what I showed you in this thing. Look at this. The mother has the recessive gene, father has the recessive, and then in order to suffer from the disease, she has to get it from both. So this statement is true.